Yes, ma'am. I think one of you have raised your hands.
Yes, ma'am. I just wanted to know
Yes, ma'am.
that whether there is any provision that if I am not able to give the exam this time, I can give it later on.
Yeah, if you haven't registered or if you are not able to give the exam, you can again enroll in the course, enroll for the exam, and then give your exams.
So that is again after 4 months, isn't it?
Yeah, again you have to enroll it. I guess this will be repeated one semester later, again next year. So there you can again enroll for this course, take up all the assignments and stuff again for the 12 weeks, and then enroll for your exams and give your exams.
Okay, okay. So no provision in between, isn't it?
No, I guess there is no repeat.
Or there is a repeat only. Okay. Okay. Thank you. Thank you so much.
Thank you. Yes, Ayushi Gurjar.
Good evening, ma'am.
Good evening.
Ma'am, I want to ask that what is the level of the question paper? Means the question paper will be moderate or hard, or me like what is the matlab level?
Okay. I honestly don't have any opinion about the exams. I have not taken an exam for this particular course. I have done other NPTEL courses. So, from my experience, I would tell that if you are going to have a good grasp of the lecture contents and the assignments, basically, you will be able to do your exams well. This is from a general perspective. I hope that goes well for this course also. uh, it's like the lectures were pretty extensive. It covered too many topics, and whatever I could cover from my part in the 2 hours every week, I did that. So, if you have a good grasp of the concepts, I think you will be able to give the exams pretty well. I Have you solved all the assignments so far?
Yes, ma'am. Yes, ma'am.
So I think that's more than enough to give the NPTEL exams.
Yes, ma'am.
Mm. So it's the level of questions depends on your understanding. So I guess you have a good grasp of the subject now, so it should be easy.
Okay, ma'am.
Any more doubts?
Yeah, ma'am. One more thing. The questions will be, as we have computer-based exams, so it will be multiple choice questions.
Yeah, it should be multiple choice questions mostly.
Okay. Okay. Thank you.
Ma'am, actually, there is any topic, matlab, there is any important topic like we have to give more focus on that.
No, we don't have a blueprint of how the question pattern is going to be, what is going to be focused on. At least I don't have any blueprint like that. My role is just to facilitate the knowledge that about the microbiology course. So I don't have any particular idea about how they have framed the question paper or how it's going to be. But important topics, maybe for this subject it's very difficult to say, at least because it covers a huge information, but all comes under something called the fundamentals. it, you need to know all of this to pursue some, maybe what to tell, some career or something related to microbiology. You will need the understanding of all the 12 weeks here. But from the exam point of view, again, you can just focus on whatever assignment questions that were given. You can see like what kind of questions are given, what kind of concepts and thought process is required for solving assignments. Maybe that should be enough to attend the exam also.
Okay, ma'am. Thank you.
Any more questions?
I have a query. So,
Yes.
is the questions will be of typing format? Any question? Because we have practiced with questions in the 2 hour sessions
Questions will be of
in the
type.
form of
So
filling the blanks.
Mostly it should be about selections only, like multiple choice questions, or it's about selecting the answers. I guess there won't be anything to type, like any reasoning question or something like that. Might be you can expect some numerical questions, even those will have options. Most probably they will have options only.
Okay, thank you.
Otherwise, you will have a blank where you just have to type the numerical value, just like we do in the gate exam. That I don't think that's the least possible thing, but maybe that should be the least thing that happens. But mostly, it's going to be selection kind of thing.
And is there any negative marking, ma'am?
No,
Okay.
uh, I'll just check because in the purview of the exam, like I am not sure about what pattern is going to be followed and stuff. That will be taken care by the course TA itself, from the faculty side, from the professor side, who's in charge of taking this course.
But madam, whatever exams I have given, there is never a negative marking in any of the NPTEL exams.
Yes, sir. So I think the same applies for this also. Unless and until something is very specifically mentioned to you that okay, there is something like that, it's not the case. Just like any other exam, this is going to have the same pattern. Any other question in the meantime?
Madam, I just want to share one thing to others. Like some of the questions are having multiple answers and some are having only one answer. So that can be made out from the circular or the square shaped, answers.
Yeah. Yes, so thank you for mentioning that. So what sir is trying to tell is some questions will be multiple choice questions, where you will have four options and you can select only one option from that. That will not have a checkbox in that. But there are some questions called multiple select questions, where one question will have more than one right answer from the four options that is given. In that, you will have a checkbox. So you, like we fill in a normal online form, if it's going to be a checkbox, you can check more than one option in that. Whereas in the case of this multiple choice question, you can select only one option. If you select a option, other options, selection of other options will be disabled. If you are going to select B, the selection of other three will be disabled. So you cannot select two options at a time for a multiple choice question, but in a select question you can choose. So based on that, you can be careful. You can just know if you have to select one or more. So that's what sir was trying to communicate. Thank you for mentioning that, sir. Okay.
And also,
Yes, sir.
rightly, you have already told that if you focus on the assignments only, that is good enough to clear the exam.
Yes, sir. Based on my previous experiences of giving NPTEL exam also, that was something crucial. So I would suggest all of you to go through all the assignment questions properly. Okay, now let me share my screen for today's session.
And one more thing, when will we be getting the solution for the 12th week assignment?
Okay, usually your assignment answers come by, maybe last assignment's answer was on 12th.
Saturday we have exam. That's why I'm asking.
Okay. I don't know if it'll be given to you before your exams. Maybe you can just try requesting for the solutions in the discussion forum, so that it reaches the right person who has to give you the answer. Who has to give you the key.
Okay. Thank you.
Can I add something to this?
Yes, sir.
Can I add something?
Yes, sir.
The moment you submit your answers at night, on today itself, you will get the right answer. The
Because
only
the
issue is
heading
regarding the
will be
numericals
overtaken.
where you need these steps.
Hmm.
Okay, okay. So it will be there in my inbox. Okay, thank you.
In case you're not getting it, you can just mail in the discussion forum so that you will officially get the key. Like as every week they post a key for the assignments that you can get by mailing them. In case you're not able to find it in your login.
Okay, okay, thank you.
I hope the screen is visible. PPT is visible, right?
Yes, visible.
Okay.
Yes, yes, visible.
Okay, thank you. Um, so yeah, for those who have joined late, I am Guru Priya, Primary Research Fellow in the Department of Civil Engineering, IIT Madras, and I'll be handling this last practice session for the Applied Environmental Microbiology course. So till yesterday, we have had 12 classes where we discussed about each week's content. Today, it's just going to be a revision of it. Uh, so this is the whole course content that this course focused on, right? Starting from a structure of the microbes till going to the applications or the environment based applications of microorganisms. We have had extensive discussions on all of them. I hope you now have a good grasp of all of this. So this is a vast content, but all of these are interrelated. You have to have a good grasp of all of these to have a very clear fundamental understanding of what environmental microbiology is. So first we studied about the cell structure, which comes under cell biology, and then we moved on to central dogma, which central dogma microbial metabolism and energetics. All of this includes your biochemistry part and the molecular biology part, and then we went on discussing about techniques to study. So those techniques to study helps us in doing an analysis about the functions, the diversity, the structure, and to know about how this interaction, the ecosystem varies, and all of it. And one important thing was discussed in week 12, which is the bioinformatics. Bioinformatics, yeah, we had to look about it, maybe not in very detail, but we saw about bioinformatics in few of the previous weeks also. So bioinformatics is a very important tool in studying this entire subject. And then coming on to the application part, since we are concerned with environmental microbiology, we went on with knowing about bioremediation, uh, solid waste microbiology, and regarding water systems also, wastewater, drinking water, and built microbiology, of course, about the structures that are around us. And then one of the major concerns of today, or the hot topic of today, is the antimicrobial resistance, where people are doing much of research to come up with new solutions to prevent it and to control it simultaneously. And in the purview of applying something, we need this epidemiological studies also. So that's pretty much. And then most importantly, we have biosensors. So these biosensors are developed based on the principles that are involved in microbiology to again help us have some applications to detect this microbiological processes or presence. So all of it have their own importance in building any system. So it's not like you have something very focused based on your research interest, but All these topics that are listed here are very important to know about anything in applied environmental microbiology. With, and also I would like to suggest that if someone is much interested in taking up a career in this particular field, based on whatever is your specific research interest, you can again go about reading all these topics in detail that pertains to your particular topic. For example, if we have this wastewater microbiology, you still have to learn about how the wastewater system works, and right starting from the cell structure till the biosensors, all these part you can study for your particular wastewater microbiology also. And even in that you will have much more specializations depending on what exactly you're going to work on, industrial waste or a municipal waste, something like that. So if you have an idea of pursuing this to your career or with your research, you can obviously do that. There are many research articles and books available, uh, but remember you just have to have a knowledge of all these factors again to ponder more in that. So that was the entire purpose of this course and the course content structured in this way. So with this, I'll just post the first question. All the questions are going to be identify the true statements. I have two or three identify the false statements or something like that. So we'll try to solve as much as questions possible. I'll wait for a few minutes to get the answer for this in the chat box. Okay, we have a handful of responses now. One more thing I would like to add here is I facilitate these sessions independent of knowing about how this course is being handled by the professor. So the questions that are here is meant uh, for your practice, just to make you understand the concepts and uh, think, or to make you think in such a way so that you can answer any such random questions about this topic. You need not worry about these questions appearing in the exam or not. Even I am not sure if that's gonna happen. So first thing is I don't have an idea about that. Next thing is all these questions are just practice questions, and I know the audience that I have are from varied backgrounds. This course is a microbiology course offered by a civil engineering department again. So, a civil engineering faculty again. So, this has its own diversity. So, in case if you are not able to have a grasp of few concepts seem new to you, I would like to say that that's completely okay, because keeping in mind that you have your exams on Saturday, now just focus on whatever concepts you understood, revise them, and whatever simple concepts that more concepts you are able to grasp, do that. You cannot read about everything in microbiology and go and give your exam. So I got two or three questions that made me feel like you are a bit tensed about what will be appearing on the exam on Saturday. Make a clear understanding that you are here to understand the microbiological concept, and based on your understanding I am pretty sure you will be able to take up the exam very well. And even after the exam you still have time to learn more about microbiology and know about it. So as far as this exam is concerned, or this practice session before the exam is concerned today, please make sure you have a clear understanding of whatever you know already. If you are able to see, this question can have more than one answer. If you are able to find at least one answer right, that's again a very good thing that you have learned from this course. So please have that in mind, and don't get intimidated by the practice questions that we have in today's sessions. And another information is all the questions that we are going to discuss today, I have somewhere discussed this in the 12 live sessions that I have conducted so far. So that's another thing that I wanted to add before answering this question. So the answer for this is B and D. I guess none of them got it right. Protozoans are not prokaryotic and they are eukaryotic. Prokaryotic ribosomes, like this sedimentation coefficient is nothing but the Swedberg units, what we call as 70S and 80S. So that S stands for Swedberg units, which is a representation of sedimentation coefficient. So in that case, prokaryotes have 70S, and sample for AFM is just a topographical study. So the samples for AFM do not contain fluorophores. Um, and then I think for confocal and all, you will have this fluorophore coming in. And then gram-negative bacteria has an additional layer of plasm than the gram-positive bacteria. So all these four questions I have discussed in all these four options, maybe as individual questions are a part of some other question, I have discussed in the um, first week live session again. If you want a detailed explanation, you can just go back and watch. This is the next question. today here in this practice session, I have all questions which reads either identify the true statement or a false statement, just because I can cover more concepts, having four different concepts as four different options. That's the only idea. But in your exams, be careful to read the question correctly. Just read if they are asked for true statements or false statements. That's one mistake we tend to do in some hurry that happens in the exam hall. And one more thing is have an open mindset. So all the live sessions that I have handled so far has been pertain to whatever that was covered in that week's lecture. If week 12 lecture is about bioinformatics, I only had questions about bioinformatics, nothing more than that. So but today we will have questions from the entire portion of microbiology. As we have already seen, molecular biology concept may be helpful in applied part, a bioinformatic concept may be helpful in the applied environmental microbiology part. So have a open view while looking up for the question. You will not know which week's portion or which slide has the answer for this. You just have to have a very open mind before reading the question, so that even if you don't know, you can relate and find answers for the question. I think in few sessions we also had questions where I told you, even if you don't know about that, how you can eliminate other options, or how you can use your understanding about other concepts to answer the given question. So please don't have a very narrow idea of knowing the terms and then getting the right answer. Just be open for giving your answers. Okay, we have B and C, A and D. Okay, let's see the answer. It's B and C. So people who have answered it B and C are right. tRNA helps in the translation process, so it's not in the transcription process. Nucleotide, yeah, when we have nucleoside attached to with the phosphate group, we call it a nucleotide. And polypeptides, because they have multiple peptide bonds, individual amino acids are connected with peptide bonds, and when we have multiple amino acids connected with peptide bonds, we call them as polypeptides. So this polypeptides then make proteins, secondary structure, tertiary structure, and quaternary structure, etc. And identify, then the last one is DNA strands are parallel and they form helix. DNA strands are actually anti-parallel, and yeah, they form a DNA double helix. So because it's anti-parallel, the last statement is false. The true statement among the following are B and C. Next question. most of you have given A as a true statement. What about ribozymes? Okay, let's look at the answer for this question. All enzymes are not proteins because there are one class of enzymes called ribozymes. They are RNA molecules and they still act as enzymes. So scientifically it's not right to tell all enzymes are proteins, and the vice versa is also not true. Not all proteins are enzymes, not all enzymes are proteins. We have something called ribozymes. And then alpha helix is a secondary structure phenomenon. Alpha helix and beta pleated sheets we have in the secondary structure of proteins, not in the tertiary structure. The tertiary structure is just three-dimensional structure of the protein is in the tertiary structure, and in the quaternary structure we have multiple tertiary structures attached together to form the quaternary structure. And we know that simple long amino acid strand, it forms the primary structure of the protein. And then yes, tryptophan has a aromatic group that it has a ring in its structure, so it's aromatic amino acid. And cysteine is one amino acid that contains sulfur. In week one session or something, I just shared a table which contained the structures of all the 20 amino acids. You need not mug up all the structures of the amino acid, but just go back to the table and know about individual amino acids, like something peculiar about that any amino acid. Only few amino acids have sulfur in them, so you can just go and have a look of those structures and have that in your mind. I'm repeating, you will not go and mug up all the 20 structures, but just get a note of what peculiarity some amino acids have. It's just two or three aromatic amino acids are there, only two sulfur containing amino acids are there. And maybe you can know the classifications, which is a positively charged, negatively charged, something like that. All these basic things, if you have in mind, that would be enough. This is the next question, and all of these options we have discussed in one of the live sessions. So please think, recollect from those sessions, and then you can answer. Okay, we'll see the answer for this question now. I guess a couple of you have got it right. A, B, and C. See, we know something called a genetic code table. So in that table, you need not mug up the codes for everything. The very key points or the highlighted points in that is that tryptophan is coded by only one codon, methionine is coded only by one codon, and that codon is called the start codon. And stop codon, we have three stop codons. We have one start codons, three stop codons. Methionine and tryptophan are being coded by only one codon. Whereas for other amino acids, we have more than one codons which can code for that particular amino acid. For some, we have four codons. For some, we have two or three codons. So that thing you need not keep in mind. You just have in mind whichever is something very peculiar in that particular topic. Okay, so with that, now you know tryptophan is coded by one codon, start codon codes for methionine. Yeah, that's true. Stop codon is a nonsense codon. It doesn't code for anything, it just stops there, right? So that's why we call it a nonsense codon. This wobble based pairing amplifies the effects of DNA mutations. No, it's not like that. See, what is wobble based pairing means, you if you want to have serine, for example, AGU also codes for serine and AGC also codes for serine. You have a DNA strand in which in one point there is a mutation in the DNA, and in the mRNA transcript that you get, instead of getting you get C. You have A G there, and in the third position, instead of, based on your non-mutated DNA, you should be getting U, but your DNA is mutated, and now you are in the RNA transcript, you are getting it as C. Even then, when you are going to translate it into a protein, it will be coding for serine amino acid only in that case. Okay, so verbal based pairing does not amplify the effect of DNA mutation, but generally, in few cases, it can reduce the DNA mutation effects, because of the same reason that more than one codon can code for the same amino acid. Okay, so that's the reason why D is a false statement. I hope it was clear. If you want me to repeat, just put it in the chat box. Okay, I guess it was clear. I'll just post the next question. If you have any doubts or if you want me to repeat, just post in the chat box at any time. I'll just wait for 1 more minute to see if there are any more responses. Okay, I guess we are good to go. Only C is right answer. I get few of you have got it right. So high turbidity, generally in exponential phase, the cells grow rapidly. So from being low turbid, the turbidity changes to become high turbid in that phase. So that is wrong in that statement. And the measurement of turbidity and plate count. So plate count is a direct method for microbial count, but measurement of turbidity again is not a direct method where we are going to count the individual number of cells that they are present. Instead, what we do is we just rely on some optical density reading, and we try to get a relationship between that reading and the number of cells, cell mass, cell density in the sample that we have. So that is not a direct method for microbial cell measurement. And the third one is extent of lag phase depends on the age of inoculum. That's true. We are going to add a active log phase culture. Your lag phase will be shortened. So age of inoculum does affect the extent of lag phase. And when the microbial population is lesser than the carrying capacity, microbial death start to occur. No, carrying capacity means, as a word suggests, how much the system can carry. So if the system can carry some 100 microbes, and if you have only 50 microbes, it can still more have 50 microbes in the system. But if the carrying capacity is getting exceeded, that if I can hold only 100 microbes but 150 microbes are present, it cannot hold it. So obviously cell death starts to occur. So when microbial population is higher than the carrying capacity, microbial death starts to occur. That's when that statement will become true. When it is lesser, yeah, it can even grow and the numbers can increase in that case. So just try to understand the statements before answering. Just have a careful look, and for once think about the key terms and the definitions of those terms. Even if you don't know what the exact definition is, you can just connect. Carrying capacity means, okay, how much the system can carry, how much cell the system can have. Okay, that will define carrying capacity. If that is going to get exceeded, then cells will start to die, because it can hold only this much, so the cell will start to die. If it's the vice versa, the cell will start to grow only. Okay, so that's one way of answering. It's not that you can know everything and then give your exams. So please be careful with questions like this. Okay, I've just included only one problem in this entire session. It's a simple problem again. In all the previous sessions that we have had discussions about problems, I got very few responses as answers. I guess you are able to answer it now, because this was the same question that was discussed in one of the previous live sessions. I'll just wait for 2 more minutes because this is a problem. So all of you have your own pace of solving problems. So I'll just wait for 2 more minutes and then I'll discuss the answer. Okay, I'll just display the answer now. So here we have this formula where if you want to know the number of cells that is present after few hours based on the number of generation, the generation time, and the initial number of cells, you can find it where x is the number of cells after n generations, 2 power n is where n is the number of generation and x naught is the initial number of cells. So here we have 48 hours and the generation time is 2 hours. So the number of generations that we can have in the given duration is 24 generations. So we have 2 power 24 into the initial number of cells that we have is the 10 to the power 4 cells. So that gives us 1.7 into 10 to the power 11 cells. Okay, so there was one more question in the assignment regarding this growth rate and all. I hope you have the solutions for that. Even that was discussed in the same this week's content. I guess you are able to access the PPTs of all the 12 weeks presentation in the Google Drive. There is one drive where I'm sharing all the PPTs of the presentations that I'm making. Even if you don't have time to check out with the videos, you can anytime go back and check with the PPTs for the questions that were discussed in the live sessions. I hope the problem was clear. If there is any doubt with the problem, you can just post it in the chat box and you can try answering the next question. Okay, we have multiple answers here. So all the statements are true. I guess you have confusion with B. Most of you have left it. So we all know electron will spontaneously move from more negative potential to more positive potentials. So that is how this difference in potential help us drive few reactions. And ATP, of course, from ADP, if you want to have ATP, you go for this phosphorylation reaction. You have this addition of phosphorus, and then NADH is a coenzyme. Nicotinamide adenine dinucleotide hydrogen is a coenzyme in reactions. The B option is that free energy is released when electrons move from a reductant to an acceptor with a more positive redox potential. So this movement is spontaneous, right? From a reductant to an acceptor, the movement is spontaneous and you have this free energy released during this process. You can just understand from the word itself, it is going to have a spontaneous movement and hence there will be release of energy, which means there is no energy consumed, but because it's spontaneous, you have this energy release here. I hope now you're clear. B is also a true statement. Just read it carefully. If you want, you can also convert the options into some diagrams and see if those works for you. Okay, so the answer for this is regarding glycolysis, it occurs in the cytoplasm, not in the mitochondrial matrix of microbes. And on the other hand, we don't have any specific organelles present in microorganisms. Basically, that is one more point which I want to discuss regarding this. So please have an idea of the difference between prokaryotes and eukaryotes that we saw in the first week. And then it occurs in aerobes as well as in anaerobes. So glycolysis occurs everywhere. After that, only if it's going to be aerobic, anoxic, or anaerobic, whatever is going to be the fate, that's going to be different. But glycolysis is common, and it has two phases: the preparatory phase and the payoff phase. So in the preparatory phase, all that happens is consumption of ATP. Only in the payoff phase we the get to have this production of ATP. But since we produce more, the net ATP production is positive in the case of glycolysis. So even if you don't know the intermediates of glycolysis in each step, just have a fair idea of what is the general backbone of this glycolysis, where it separates into two, and how much of ATP it is synthesizing in individual steps, how much it is consuming in individual steps, that how much is consumed, how much is produced, and what is the net ATP production with glycolysis. So I guess you can go through that once. So here is the next question. This is a mixture of many questions that we have discussed in one of week 7 or so, something. The only intention was to prep, brush up with all the concepts that we have gone through so far. Okay, two people telling all are true. So anyone else who wants to answer? Okay. Okay, we have a variety of answers here. Three people telling all true, AC, CD, and only C. So from all of it, what is clear is C is surely true. Okay, any of you remember what is anammox or the expansion of anammox basically? Just that one thing. If any of you remember, can you just give in the chat box? Hmm. So it's true, right? Okay. Okay, we'll see the answer for this. The only true statement is that anammox stands for anaerobic ammonia oxidation. So it is a nitrogen removal technique for wastewater treatment. Energy yield is more when electron donor is inorganic. No, it's the other way. Energy yield is less when the electron donor is inorganic. See, we have, I think, 2 or 3 times I've discussed about aerobic, anoxic, and anaerobic conditions, right? So in aerobic, what happens is terminal electron acceptor is oxygen, and when the electron donor, when you're going to have some carbon based, you will have the energy yield as high. Whereas in case of inorganic, your process is not going to go until a longer electron transport chain because of the constraints that are based on the electron tower. Okay, so in that case your energy yield will be less because your starting point is an inorganic compound. If it's an organic compound, the energy difference will be somewhat more, so you will get more energy yield. So that statement is false. Anammox is a nitrogen removal technique. Yes, enteric bacteria are pathogenic. Not all enteric bacteria are pathogenic. Enteric bacteria, I guess you remember that it means it's something related to the gastrointestinal tract or the gut basically. We have beneficial organisms also. Not all enteric bacteria are pathogenic. Facultative, as the name indicates, they are able to facilitate between something. Obligate in the sense it's strict requirement. Facultative is not like a strict requirement, right? So in that case, facultative anaerobes can survive in the presence of very very mild oxygen conditions. But if it's going to be some strictly anaerobic microorganism, even if you give very very trace amounts of oxygen, it's going to be toxic to that organism and it's going to die. So this statement was based on the difference between what is facultative and what is obligate. Facultative, as the name suggests, has some tolerance and can survive until a certain limit. So, I have not mentioned what is the threshold of oxygen that I'm going to supply to the anaerobe. So, in that case, this statement is false. Facultative anaerobes can survive in the presence of at least a mild oxygen. I hope it's clear because I got a lot of wrong answers. If you have any clarifications or discrepancies, you can just post in the chat box. In the meantime, I'll just display the next question. Okay, this question was also discussed earlier. Please note the question. I have asked which of the following statements about redox potential is false. Okay, let me just display the answer. So A is the answer. Uh, NAD plus NADH redox pair does not have the highest redox potential, but in case just the fact that oxygen and water redox pair has the highest redox potential. So this is just memory based. If you know the electron tower, you can answer this. The next point is that components of electron transport chain are organized in terms of their redox potential. That statement is also true. So that's why we get a difference in potential, and ATP synthesis again happens. And then redox potential of our system is usually compared with the potential of hydrogen electrode. Yeah, the reference for any redox potential measurement is with hydrogen electrode, and that has 0 as its potential. This is all facts, so I guess you are clear about the facts. Now we'll go to the next question. So until this, we have seen about all the basic cell biology, molecular biology, biochemistry, metabolism, energetics, and stuff until that question. So it was a mixture of all those topics I've noted down. I see someone raising their hands. If you have some question, you can just ask. Okay, I see Sony P has raised their hand. If you have any particular question, you can unmute and ask or put it in the chat box. And others in the meantime can try answering this question. Okay, I guess we can discuss the answer for this question now. So, ion torrent semiconductor sequencing exploits pH differences. We have discussed this already. pH difference is the key aspect in ion torrent semiconductor sequencing. I think this was also discussed in one of the lectures. And then gene transfer from one bacteria to another, that is not its offspring, is horizontal gene transfer, not vertical gene transfer. Conjugation is a horizontal gene transfer. It's not like it happens between the offsprings. It happens between two bacteria of almost, which can be called as of the same generation. And then fibromouracil is a DNA base analog. I hope you remember what a base analog is. So when we studied about the mutagens and the mutations and the mutagens that cause the mutations, we studied about radiations. We studied about what are the mechanisms that can happen. So in one case, fibromouracil, which is a uracil only, but with a bromine group attached to it, can act as a base analog, which means that it can have its position in the nucleotide strand, but since it's bromouracil, it cannot be translated effectively. So that in that case, it is a base analog, which is analogous to the base that has to be present in that position, but causes a mutation. We have also seen about intercalating agents like acridin, if you remember, and also about UV radiation. So UV radiations, make, they facilitate the formation of thymine dimers, and by that case they act as this mutagens. So, and in the case of horizontal gene transfer also, horizontal gene transfer means transfer of gene from one bacteria to another, in this, in the so-called same generation. If it's going to be from one generation to next generation, then we call it as a vertical gene transfer. Okay, so in horizontal gene transfer we have this conjugation, uh, and we also have transduction, and we also have transformation in that. So I hope you remember all these concepts. So here is the next question based on the applications part. Okay, so all of you are right with the answers. Viruses are counted by PFU, bacteria are counted by CFU. Ruminant animals, yeah, they're capable of digesting cellulose. And biomagnification is not a prerequisite for bioremediation. So, and then commensal bacteria are not found only in biofilms, they can be found elsewhere also. So, for now, I'll post the last question for today. Again, a easy question. Again, all these were individual questions in one of the sessions. I've just clubbed all the options together. Okay, let's just see the answer for this question. So, anti-protozoan drugs, because protozoans are eukaryotes, there are chances that human cells are also eukaryotic, so it might happen. So, that statement is true. Polyoma viruses cause tumor. Retroviruses are a group of viruses which have something called reverse transcriptase enzyme. So, transcription process we all know, which is DNA to RNA, and the reverse of that process is facilitated by the reverse transcriptase enzyme. So these viruses have the capability to shift from DNA to RNA, RNA to DNA. So from a medical point of view, where this happens to be a problem is that um, if you're going to target a drug for any diseases that caused by the virus, uh, it will be very difficult to target either DNA or RNA because it has this capability to shift between these two. So maybe that you can have in mind. Enteric protozoa require vectors? No, not always. So enteric protozoa, the infection is going to be by ingestion. So I think because of that they don't need vectors. Okay, so because it's a gastrointestinal problem or something, they don't need vectors. Okay, so I just have few more questions also in this PPT, which is again based on whatever we have discussed in previous sessions. So I don't guess there is nothing much new in this. I'll upload this in the Google Drive. You can just go through this PPT once. I'll do it right away today night. I know you have your exams on Saturday, so I'll just do it right away today. You can just go through the other questions also. I think I have. 4 or 5 more questions, so you can just go through them when I upload it. Now I am just leaving the session open to address any specific technical doubts that you have, any doubts that you have. I also had a discussion in the start of the session. In case some of you joined late or something, I'll again give you a few minutes, and for now I'll just stop presenting. Okay, yes sir, I'll give it. I hope the screen is visible. So here you can check out, sir, in the slide that I'm presenting. We have typhoid, which is a waterborne disease, dysentery, which is a water-washed disease, schistosomiasis, which is a water-based disease, and malaria, which is a water-related disease. This is from week 9 discussion. You can take a screenshot of this so that you can go back anytime. And here is the definition of what is waterborne, watershed, water-based, and water-related. Okay, I guess it was pretty much clear, and I hope no one else has any doubt. So in that case, if you don't have any other questions or any feedbacks or suggestions, thank you so much for joining in for this session. For any of the previous sessions and including today's sessions, it was very good interacting with all of you. You were really very attentive and interactive because all my sessions which were based on questions, so unless and until you answered, it couldn't have progressed more. So thank you so much for being patient as well as interactive. I wish all of you all the best for in case you're taking up the exams. Thank you so much for joining in.